Okay, you guys. So we left off. We talked about the miRNAs that can either degrade the mRNA or physically block transcription. So then we just have the post-translational modifications. And so oftentimes when transcription happens and you get a chain of amino acids, this polypeptide, it's not in its finished form yet. So it doesn't have the right shape and so on. So oftentimes we have to have modifications to cause that to happen. And so the types of modifications are cleavage. This is the first one, cleavage, which means that you can cut off certain parts of it and so on, so that that'll affect the way that the polypeptide bends and bonds and so on. The second thing is we can chemically modify it by adding certain chemical groups to it that will also alter the way it bonds and therefore alters its three-dimensional structure. That chemical modification, this occurs in the Golgi apparatus. And then this says transport to cellular destination. The transport itself doesn't modify it, so therefore the third thing is this last, after the protein has become active, the last thing after translation, the third thing is the degradation of the protein or the breaking down of the protein. The purpose of this is so that not every single protein that your cell has ever made stays active for your whole life of the cell. So if that's the case, then you wouldn't have no control over what the protein does or not. And so that's how we can control certain things in our cells, either make the protein and have it available or not. And so therefore, the protein eventually does get degraded. And so this degradation of protein is caused by adding a certain chemical to the protein. It's kind of like the tag that says, destroy me, for the protein. Does anybody know what it's called? Ubiquitin, yep. So we add ubiquitin to the protein. And as I said, that is like a signal for destruction. And then another molecule comes in to actually destroy the protein. That starts with a P. Proteasome, yep. So you add ubiquitin to the protein, and then a proteasome is what degrades the protein. So that's your last thing that is helpful in regulating gene expression in eukaryotic cells. All right, so what I'd like to do next is relate this now to the article that you read yesterday. And so the title says, genes hold the secret to survival of Antarctic antifreeze fish. And so early on in the article, it talked about how these fish are able to live in cold, oxygen-rich environment. I mean, cold water, oxygen, there's more oxygen found in dissolved oxygen that's dissolved in the cold water than in warm water. And so that's just characteristic of cold and warm water. The oxygen is O2 in the atmosphere, the same oxygen that we breathe. When it hits water, it can dissolve in it, and so that's dissolved oxygen, and that's the oxygen that fish use. And they can use that dissolved oxygen, taking in through their gills and so on. So the fish aren't using the oxygen in H2O, all right? They're using O2 that's dissolved in the water. And because cold water, there's less movement, all right? So it slows down the movement of molecules, so less oxygen actually leaves the water. So there's more oxygen in this cold temperature water. And so it said very early on that these fish manufacture their own, quote unquote, they call them the antifreeze proteins. And so what do these proteins do? Lucas? Say it out loud so people can hear you. So I can barely hear you, so I know that these guys can hear you.
They bind with like ice crystals in the blood that was formed and the blood is freezing to like liquefy them, or they just
spaced apart, so yeah, all right. Yeah, so that they don't freeze.
She's also sending you an email, but
Okay. Okay. Who is this?
Ms. Schneider.
Yeah, that's Paula. Okay. So so that absolutely. So they prevent the fish from freezing, and so they studied these genes in gene expression. They looked at different tissues of the fish and so on. What else did they discover about them? Matt?
There's more about them in the fish in Antarctica because of duplication.
That's right, they're more abundant in the fish in Antarctica than in warmer waters, and they attributed that to gene duplication, which I talked about at the end of chapter 19. So gene duplication is when you have a gene that is duplicated so that your DNA on the DNA gets more than one copy of that gene. So everybody remember how duplication occurs?
They crossed over like the wrong way, so the corresponding sites for crossing over are basically not alike, or at least so more part of one non-sister protein goes to the other, and one part goes to the other. So you have duplication in one and deletion in the other.
Okay, absolutely, absolutely. And so, when that occurs, you can have out of your two homologous chromosomes, one that has two copies of the gene, and one that doesn't have a copy at all, because of that like unequal crossing over. So it's kind of like if my arms were the chromatids that are crossing over, and remember when crossing over occurs, you're supposed to cross over at the same point, so like at the wrists, and so everything exchanges from the wrist to the ends of my fingers, and I'd switch hands. Remember that when crossing over? So unequal crossing over is when, like, where one, let's say my elbow and my wrist, and this is where crossing over occurs. So now when crossing over occurs from my elbow all the way to my hand, will be put on this part, all right? So I'll have two parts here, all right? So therefore I have two copies of all those genes. Does that make sense? And the other one that was like this would just have my hand here, so it won't have any of those genes, all right? It'll just like a little hook to my hand, all right? And so that's what that would look like. And so you get gene duplication. And so what they found is that these cold water fish, they had lots more genes than the warm water fish, attributing to gene duplication. How does the having multiple copies of this gene or these genes help the fish?
They're able to produce more antifreeze proteins.
Yeah, so they are able to produce more antifreeze proteins, so therefore help them to therefore survive in the colder water. All right, and so so that's basically what they did. And so remember also one other thing about when genes duplicate like this is that it's possible. So then if you get multiple copies of genes, it's possible that the gene, like this is the original gene, so this is a duplicate, that a mutation could happen in this gene that would produce a different protein or a slightly different protein. And so you can get families of genes that are closely related but produce slightly different proteins. So I gave an example in the notes about the globin protein family, like hemoglobin is an example of one of those that we have, and I showed you two different chromosomes in humans, that we have these families of globin proteins that they're very similar in that they produce these globin proteins like hemoglobin to bind oxygen, carry oxygen to our cells, and they think that those genes originated from one gene that has duplicated and had slight mutations in them. And so this would be an example with the antifreeze gene. It also talked about that they found that that it produces proteins that protect them from extreme cold and heat. They also found that in the fish that lived in the cold water, that they produced a high amount of ubiquitin compared to warm water fish. And remember, ubiquitin is the destroy me tag on the protein, and so the proteins are destroyed. Why that is helpful is that it helps the fish to maintain health by tagging proteins that there's something wrong with them, damaged proteins or something like that, and destroying the damaged proteins and helping them to stay healthy. And so that's another way that this relates to chapter 19 that they talked about, that it expressed in there. All right, and so the antifish protein in this antifish gene is one example that the College Board specifically gave us AP Bio teachers. This is an example of something that you need to know as far as how this relates to that. All right, so it's a real life example of some of these things. All right, so I'm going to have you pass forward your summary. Make sure your name is on it. If you wrote your summary on a separate sheet of paper, I only need your separate sheet of paper. If you wrote it on your article, you have to say your article.
What book are you reading? Be a civilized person.
Okay, so what we're going to do is take a look at and relate what we just talked about with genes being expressed or not expressed in different types of cells and the things that cause that to happen. We're going to relate that now to basically when this occurs in us, in multicellular organisms, and that's very early on in development. So as very early on in development, during embryonic development, that's when cells become specialized and get predetermined their fate, and so on. So that's what we're going to look at here. So we all start out being one cell big. Sperm fertilizes the egg. There's your zygote. And so there's three major changes that occur in the growth of the organism. Very basically is cell division. So that we've talked about with mitosis. I said, okay, we start out with one cell big, then we divide and become billions of cells big. But there's way more to it than that, because we just don't become billions of cells big and be a big blob of cells. We actually have the cells become differentiated, and also we get shape and form. And so we're going to look at how do we know the head and the tail, the front and the back, and the left and the right, and so on. All right, so cell division at first. This is a three-dimensional picture here. So you start out, this is the zygote. They're skipping a couple divisions here. It'd be 2 cells, and this is the 4-cell stage, and so on and so forth. And so very early on in development, the cells look pretty close. In terms of physically, you don't see specialization occurring right away. But we're going to learn that right away, actually, in the first cell division, that when that first cell division happens, while these 2 cells are genetically identical, because you have 46 chromosomes and they make copies of them, and then each 2 cells with 46 chromosomes, so genetically they're identical, but we're going to see that actually the molecules in the cytoplasm are not identical. So very early on in development, with that first cell division, actually starts the process of differentiation. And so then eventually we get to cell differentiation later on in development. The cell differentiation is the process by which the cells become specialized in form and function. And these cells undergo changes that organize them into tissues and organs. And this picture here just shows you a couple of changes that occur very early on in development. So this is the surface, what they look like on the outside. These arrows are showing you which way cells move. You can see here, this says blastula, and then this is time. So going down here, this is time. And so this is earlier on in development and later on in the development. But you can see here that each one of these kind of like little circular things are a different cell. So this is multicellular, but it still, from the outside, looks like a ball. But realistically, it's not just a ball, and you can see that some things are going on in the inside. So there's a space here, so you have some hollow space, whereas this part is filled in with cells. Mine is color-coded. The color coding is supposed to show that different cells are going to eventually differentiate into different parts of the body, so they're going to end up as different fates. And then as time goes on in development, so this is called the blastula stage, and then eventually it forms into what's called the gastrula stage. But what I want to show you is that these cells move around. So therefore, the balls of cells move around, and they're not stationary, and eventually start to form the shape and so on of the organism. So this is very early on in development because it looks like a ball still. Eventually it'll form the shape of the organism. And so then the third stage is morphogenesis. As the dividing cells begin to take form, they are undergoing morphogenesis, which means creation of form. So notice that this starts to happen as the cells are still dividing. So these morphogenetic events lay out the development very early on in development as cell division, cell differentiation, and morphogenesis overlap. So it's not like, what that means is that these things can occur simultaneously. It's not like all the cell division occurs first and then all the differentiation and so on, they could be occurring together. So these events that happened for the organism to change shape tell the organism where the head and the tail are, what is the front and the back, and the left and the right. And as time progresses later on, the later morphogenic events will give instruction as to which, where appendages are, like arms and legs and so on and so forth, and will give shape that way. But that's not until later on. And so, so these morphogenetic events, the taking shape, as well as cell division and cell differentiation, take place in all multicellular organisms, plants and animals alike. Morphogenesis differs in two major ways, though, in plants and animals. So in animals, which we'll focus the rest of the chapter on, in animals, movements of cells and tissues are required for the transformation of the early embryo into the 3D characteristic form of the organism. So movement, so that's why I was just showing you that the cells and tissues early on, like with the blastula and gastrula, that picture I just showed you, those cells are moving to help to eventually form the shape. Are we good? See, do I need to go back? Am I good? No. Okay. I haven't read it out loud yet. So, yeah, I know. I gotta change that. I should put this in black, huh? In plants, morphogenesis. I'm going to read it out loud. In plants, morphogenesis and growth and overall size are not limited to embryonic and juvenile periods. They occur throughout the life of the plant. So, in animals, when early on in development, early in the embryo, this is when this happens, where we morphogenesis and so on happens, and then it doesn't happen once you get to the adult stage, except with a few exceptions. In plant cells, and in plants, it can happen throughout the life cycle. And so let's look at this and I'll explain that. We good? Okay. All right, so here we have animal development. Let's look at this first. This is a starfish. So the starfish starts out being a zygote, fertilized egg cell. It begins to divide. Here's the 8-cell stage. And then notice here, so that's the cell division. So it's becoming more cells big. But then as we go on in embryonic development, you can see there that it just doesn't keep dividing and being just a ball of cells, that the cells actually start to form a shape and so on. So that's the morphogenesis starting. And so we see here the shape, and so it's moving, the cells are moving, and so on. So you get this infolding here. This eventually forms the gut of the organism. So these cells will specialize into cells that will form organs of the digestive system and so on. And then eventually, it doesn't show very well, I don't think here, because it has a big leap from the gastrula to the adult. All right, so that's a big leap from this to this. But what has happened here is cell differentiation. These cells have differentiated into become specialized cells for the starfish. And so once you get to the adult animal, with the exception of a few types of cells, that we don't get differentiation anymore in our cells. So for instance, like a skin cell can't differentiate into something else, all right? It is what it is. We do have some cells, we have some stem cells that, like in our bone marrow, that produce different types of blood cells, and those continue to divide and differentiate through an adult life, but that's the exception. Most of our cells are kind of stuck where they're at.
Could stem cells be injected, and then, like, so say you have, like, I don't know, a part of your ear is gone, and you get an injection of stem cells, and, like, technically could that grow back as, like, cartilage if they
Not an injection of stem cells, but what they can do is trigger stem cells to become cells of the ear. They're working on growing that in the laboratory, and then using those cells and then planting, and like, so it'd be like almost like a tissue transplant.
Oh, okay,
kind of
so
thing,
like
you know?
after the-
Yes, all right, absolutely. Okay, where the injection of stem cells, that would be like in the bone marrow transplants.
Okay.
All right, that would be, so you're giving a person, you know, bone marrow that will produce blood cells. Okay, so then in the plant development, so we get the same kind of thing going on here. So zygote to the adult plant. Plants, notice here they talk about this thing called an apical meristem, and there's one at the top of the plant. There's ones at the top, they call it the shoot apical meristem, and one's at the bottom of the roots, and that's where division continually occurs throughout the life of the plant, and those cells can divide and differentiate through the whole life. So these cells of the apical meristem throughout the life can divide and differentiate and cause new shoots and new leaves and stuff to grow throughout the life of the plant. And so they can do that, and so they get cell differentiation throughout the plant. And so we'll talk a little bit more about that later on, but like some of you may have you ever taken like a cutting, like taking a little branch off of a plant and put it in the water and it grows roots? I don't know if you've ever done that. So that's an example of the cells being able to differentiate into, you know, the cells of the stem can differentiate into the root cells. All right, so we'll talk more about that later on. All right, so that's a difference between plants and animals. So this is the exception for animals. So the ongoing development in adult animals is normally restricted to a generation of cells that need to be continually replenished. So example would be blood cells. So I talked about the bone marrow making different types of blood cells. Skin cells are an example. Cells lining your intestine, you have constantly food moving through there, and you know, cells being sloughed up and continually replenish those. So, so those are exceptions to this ongoing development in adults. Okay, so let's build on this multicellular organism. The cells of multicellular organisms come almost from entirely different differences in gene expression. So this is referring now back to chapter 19. So we have regulatory mechanisms turn genes on and off during development. These regulatory mechanisms are what makes cells different, because nearly all cells have the same genetic complement. So they have the same DNA, And so therefore turning on and off these genes and these regulatory mechanisms that we just reviewed from chapter 19 are important in this. So we know about cell division, so let's look at cell differentiation and how it plays into turning genes on and off. So there are two major things telling a cell when and how to differentiate. First one is the stuff. This is the chemicals, the molecules found within the egg at the time of conception. Conception is when fertilization occurs, and the environment in which the embryo develops. So we're going to see here that mom's egg cell is key in the proper turning on and off of certain genes and the proper development of the baby. And so that's what we're going to look at first. So the stuff in the egg. So what's in this egg cell to do this? So the egg cell's cytoplasm contains RNA and protein molecules encoded by the mother's DNA. So the mom makes these proteins and has mRNA and so on. The mRNA, proteins, organelles, and other substances are scattered unevenly throughout the cytoplasm of an unfertilized egg. These maternal substances influence the course of early development and are called cytoplasmic determinants. So key here is that they're scattered unevenly throughout the cytoplasm of an unfertilized egg cell. So we're talking about the egg cell before anything happens. There's no sperm to fertilize it, so there's no baby yet. It's just unfertilized. And so all the stuff in the cell that's not mixed evenly is ultimately going to affect the baby. And these molecules are called cytoplasmic determinants. So let's look at cytoplasmic determinants. Following fertilization, so sperm comes, fertilizes the egg. Mitotic divisions distribute the zygote cytoplasm into separate cells. The zygote divides into two cells. The nuclei of these cells are subjected to many different cytoplasmic determinants. What has been received will determine the developmental fate of each of the cells. The developmental fate of each of the cells. These help to create, cytoplasmic determinants help to create eventually an animal's 3D arrangement before morphogenesis can shape the animal. So it's going to start to kind of figure out the fate of like what's going to be the head and the tail region before they actually start forming into a head and a tail. And so I'm going to come back to these and I'm going to have you go to the next page and look at this picture first. Because this picture here shows you what we just wrote about with the cytoplasmic determinants. So this picture we divide it in half here. So A here is the cytoplasmic determinants, and B is induction, which we're going to look at, which is what we just skipped here. So we're going to just look at the top here first. So what are we looking at? So you have an unfertilized egg cell and a sperm cell. Let's focus on the unfertilized egg cell. So inside here, inside the egg cell, you have the mother has DNA that codes for different cytoplasmic determinants. So the egg cell has 23 chromosomes, and in the sperm cell has 23 chromosomes, right? So that when the zygote's formed, it has 46. And this DNA codes for these cytoplasmic determinants. This particular egg cell has 2. They're using just the circle and the triangles to show that they're different molecules in the cell. But notice here that the left side has mostly the red circles and the right side is the green triangles. So that's where they're unevenly distributed throughout the cytoplasm of the egg. So why does that make a difference with the baby? So then the sperm cell comes and fertilizes the egg cell and you get your zygote. Remember when the sperm cell fertilizes the egg cell, the sperm cell basically is a chromosome donor. So therefore, the sperm cell gives the 23 chromosomes, so the zygote now has 46 chromosomes in it. But everything else, like all the cytoplasm, the mitochondria, the organelles, basically come from the mom and her egg cell, not from the dad's sperm cell. And so therefore, if you look at the composition of the zygote's cytoplasm, it's pretty much the same as the egg cell because of that. So now you have this unequal distribution of the cytoplasmic determinants in the zygote. So now what is that zygote going to do? It's going to divide by mitosis. And so now when it divides by mitosis, notice here that the cell on the left has mostly the circular cytoplasmic determinants, and the cell on the right has mostly the triangle cytoplasmic determinants. What do you think that has to do with development? So right away, this first division, these cells, I want to point out here, since I did the chromosomes here, when this zygote divides into two cells, it makes a copy of all 46 chromosomes. So these cells, genetically, their 46 chromosomes are identical, but the stuff inside the cells are not. Why does that make a difference, do you think, Nathan?
Would that be kind of related to what we did with the liver and the eye cell, where like those cytoplasmic determinants can act as like receptors, I guess, or just proteins that can affect which genes are expressed in other places?
Absolutely, absolutely. So these different combinations of cytoplasmic determinants can turn on or on different genes, all right? And so therefore, in this cell, different genes will be turned on than in this cell, and therefore this cell will develop differently than this cell, all right? So that happens just right away in that first cell division. And this only shows you two cytoplasmic determinants, but there's many more than that. So, I'm going to put over here kind of what's going on with this picture here, so that when you look at it later, you can remember what it means here. So here, the egg cell is heterogeneous. What does that mean?
It's composed of different substances, like two different cytoplasmic.
Yes, it has so it's composed of two different cytoplasmic determinants. So it's heterogeneous and has an unequal distribution. I'm just going to abbreviate cytoplasmic determinants. So because of that, the zygote, that's also true of the zygote. So then when the zygote divides, what does that cause to happen? You end up with different cytoplasmic determinants in your cells after the zygote divides. So different cytoplasmic determinants end up in the embryonic cells as the zygote divides. So that's what we have here. So the cell on the left has mostly the circle ones, and the cell on the right has mostly the triangle ones. And how does that lead to or play a role in development? Each cytoplasmic determinant turns on different genes. And turning on different genes will eventually lead to those cells becoming different, having different jobs, all right, as a result of that.
I know who that is. Please back. Yeah. I'll laugh.
Yeah.
But yeah, I mean, she was in the drama club, and you could hear her through like two dressing rooms, like through the walls.
So this is why, when we talked with chapter 19 with activators and so on, in another class I had a student ask, well, when we talked about the liver cells versus the lens cell, they asked, like, well, how does the liver cell get a certain combination of activators and the lens cell get a different combination of activators? How does those genes get turned on and off? And this is what they has to do. All right, so different cytoplasmic determinants from the mother will turn on and off different genes. Okay, so let's go back to the part that we skipped, the environment. All right, so it's contagious. The environment in which the embryo develops plays an important factor in the outcome of the developing organism. So, I'm going to pull in some things from chapter 11. So the surface contact of cell-to-cell interaction helps to signal development. So we're going to look at cells next to each other. By the process of induction, an embryo's genes signal the expression of proteins that cause changes in nearby target cells. These signals send a cell down a specific developmental pathway, inducing, I almost have to go over there to see what's so funny, inducing further differentiation of the many specialized cells within the new organism.
You just open the door and see
Yeah, the people watching the video are probably like, what is she talking about? Probably can't hear it. Alright. Alright. Want me to read this one more time?
Yeah, I can't see
Alright.
that.
These signals send a cell down a specific developmental pathway, inducing further differentiation of the many specialized cells within the new organism. And so that's the second part of this picture here. So that's this part here. So let's look at this. So you have this embryo, not quite as early on. So this is an embryo that's 32 cells. And what we're looking at is the adjacent cell. So that we're looking at, when we look at this picture here, the top of this cell here and the bottom of this cell here, and where they meet. And so what these arrows, these up arrows from these cells at the bottom are trying to illustrate is the cells at the bottom are communicating with the cells right adjacent with them. How do cells communicate? How do they talk? They release ligands, right? Signal molecules. And those signal molecules, how does the other cell hear them? Those signal molecules bind to a receptor, right? So that's what we have here. So these cells at the bottom release a signal molecule. Now, why do the cells at the bottom release a signal molecule and not the cells at the top? It's because the cells at the bottom and the top have different combinations of cytoplasmic determinants. Different genes are turned on and they make different proteins. So this one at the bottom makes a signal protein. This one at the top makes a protein that gets deposited in the cell membrane, and that's the receptor. And so this cell at the bottom releases a signal molecule that binds to the receptor on the cell at the top. What does that do? Triggers a signal transduction pathway. If you remember, a signal transduction pathway could be like a phosphorylation cascade where one molecule activates another molecule, which activates another. And then if you recall that at the end of some signal transduction pathways, the molecule that gets activated at the end can be a transcription factor and can turn on certain genes. And so therefore, certain genes get turned on in this cell, and therefore that will help this cell differentiate as a result of the adjacent cell signaling. Question?
So in the first picture, where it's like, there's in reality, there's not just two different cytoplasmic determinants. There's like
there's lots and lots of cytoplasmic determinants. Yeah, that's a good point. For simplistic sake, they just showed 2 here. There are hundreds and hundreds
So
of
like
them.
in the first like cell division, there's gonna be like a ton of different ones.
Yes, that's right.
Concentrations will be different.
Yes, yes. And you have to also, yeah, absolutely. And so then as it continues to divide, another cell might have a higher concentration, all right, and so on and so forth. Absolutely. And so, so therefore, this is induction where one cell of the embryo communicates with another cell of the embryo and triggers genes to be turned on there. So, so the end result here, the end result is certain genes are turned on. And how do they get turned on? Because at the end of that signal transduction pathway, a transcription factor was activated. So the end result, certain genes are turned on because of an activated transcription factor. And so that will help these cells to differentiate as well. So we have the cytoplasmic determinants, we have cell-to-cell communication with that. All right, what I'm going to have us do, so we're at this picture here. I'm going to go back because there is some space underneath here. I forgot to put one thing in, I totally skipped a section there. I don't know how that happened. So go back to underneath where we wrote about number 1, the stuff in the cell, and 2, the environment. If you go back, that was underneath cell differentiation. All right, so I'm going to have us put another category, so another heading. We're going to call that determination.
Should that be a 3 next to it?
Nope, it's not a 3 because it's not underneath cell differentiation. It's a different process. So you see how cell differentiation, there was those 2 things, and so those 2 things. So you're going back out, kind of like an outline format.
Okay, yeah, I see.
Okay. So you need to write this definition of determination. It's the point at which an embryonic cell is irreversibly committed to becoming a particular cell type.
See what's going on.
Whose class is that?
Play this. It's a math study, so basically, you know.
Yeah, they're sitting on top of the
It's actually my class.
All right. So, and the second bullet underneath there was C figure 21.10, and that's the figure you picked up here just a minute ago, or many minutes ago at the beginning of the hour. So I want to talk a minute about this. And so there comes a point in time, and when when an embryo is developing, that the cell, enough changes occur inside the cell, enough molecules inside the cell and genes that have been turned on inside the cell cause it to not be able to go back anymore. So for instance, if certain genes are turned on, in this example here, in a cell to become a skeletal muscle cell. So a muscle in your cell in your skeletal muscle that contracts and moves your skeleton. And so there comes a point in time that certain genes are turned on, and therefore it will automatically develop into a skeletal muscle. And so let's look at this, a timeline here on the left here. So here's your embryonic precursor cell. So this cell has the ability to become other types of cells. So it can become a muscle cell, but it could become a cartilage or fat cell. And so what matters is what genes are going to be turned on in this cell to determine that it's going to become a skeletal muscle cell. So what happens is then the embryonic precursor cell, if it's activated by signal molecules from other cells, kind of like that induction process, genes can be turned on. So let's look at these genes. You have this master control gene, and then you have other muscle-specific genes, which means that these genes are turned on in muscle cells to allow it to develop into muscle cells. So every cell has these genes in them, but they're just not turned on in every cell. So how does this embryonic what happens when this gene is turned on? So this signal molecule turns this gene on. What does this gene do? This gene makes a transcription factor. So that means that it can turn on other genes. So once this gene is turned on, this master control gene, this cell now begins making these transcription factors. What do these transcription factors do? Notice that the cell looks still like the embryonic precursor cell, but inside it's changed because this gene has been turned on and it's now making this transcription factor. The transcription factor in a muscle cell, that transcription factor acts as it turns on other genes. So this gene gets turned on and makes another transcription factor which turns on other genes that make proteins, myosin, actin, other muscle proteins, and blocks the cell cycle. Muscle cells, adult muscle cells, they don't divide, and we'll talk about that in just a second here, but it turns on these other genes to allow the cell to develop into a fully differentiated muscle cell. And so this cell right here, once this master control gene has been turned on and it starts making the transcription factor, this cell is called, it's gone through determination, it's determined, meaning that this cell has no other choice now but to develop into a skeletal muscle cell. It can't go backwards and become another type of cell. The reason why, this gene has already been turned on, they're already making these transcription factors that are going to eventually turn these genes on and cause it to become a muscle cell. And so, so early on in development, at some point, your cells become destined to become one type of cell or the other, and you can't go back from that. And so, so these myoblasts, eventually, after all these genes are turned on to get a fully differentiated muscle cell, these myoblasts fuse together. Those people who have had anatomy, if you recall, a muscle cell is very long, sometimes it's called muscle fiber, and it's multinucleated. It has many nuclei in a muscle cell, and that's the reason why is these myoblasts fuse together into one long cell, and you have multiple nuclei in one cell. And so that happens as well, and so then early on in development, so that then they become the cell that they're supposed to because of the genes turned on. All right, it looks like we're rounding off at the end of the hour here, so we'll pick up this on Monday.